In this chapter, we're going to look at understanding race. The biology content we'll learn is speciation and macroevolution. As we go through our lecture, be sure to recognize the learning outcomes and review them. Find them in your notes and in your textbook as well. It was one of the most memorable images of the 2016 Summer Olympics. Simone Manuel, a swimmer for the United States who was considered a long shot to earn a medal in the 100-meter freestyle, turns to look at the scoreboard and realizes with shock and jubilation that she had won a gold medal. To many, Manuel's triumph was more than an individual victory. History will remember her as the first African-American woman to win an individual gold medal in swimming in the 120-year history of the modern Olympic Games. For decades, African Americans have achieved at the highest levels in the events of the Summer Olympics, alongside athletes of every other race. But until very recently, African Americans have been conspicuously absent in swimming events. Even now, when African Americans make up approximately 12% of the U.S. population, of the more than 178,000 year-round competitive athletes who identified their race on the USA Swimming Membership Form in 2015, fewer than 4,000 listed Black or African American. That's only approximately 2% of the total. Why do so few Black men and women compete in this sport? Manuel's success undermines any misguided argument that Black women and men are at a natural disadvantage in swimming. Manuel acknowledged old misconceptions at a press conference after her win, saying, "The title, Black Swimmer, makes it seem like I'm not supposed to be able to win a gold medal, or I'm not supposed to be able to break records." She credited a list of earlier African-American Olympian swimmers, including Cullen Jones, winner of gold medals in men's swimming in the 2004 and 2008 Olympics, for inspiring her to stay with the sport in which she is an outlier. It's clear from her statement that the misguided argument that African Americans were at a natural disadvantage at swimming had an effect on Manuel's confidence, despite the argument having no grounding in fact. The argument had that effect because it fits a pattern of misunderstanding that's relatively common. That is, the belief that there exists a number of innate differences among human races. Over the past several decades, biological research has examined the evidence for these supposed innate differences and found it weak or non-existent. What this research has established is that beneath a veneer of external differences, humans are all basically the same. All humans actually belong to the same species. Before we can understand the concept of race, we need to understand what it actually means to be of the same species. Species is a primary classification group with the most closely related members. It said if you are a member of the same species, you are so genetically similar to those individuals that you're able to successfully interbreed with them and create viable fertile offspring. As for the classification part of species, it's part of the binomial name created by Carolus Linnaeus in the 1700s that's given to every species so that every scientist around the world can understand what organism you're talking about. It's usually in Latin because Latin is a dead and unchanging language and doesn't have a lot of slang involved with it. So a binomial name is a two-part name, generally the genus and then the species, and sometimes a subspecies as well. The genus is a broader group that has different species that belong to it. Generally, we capitalize an individual's genus. And then the species is the second part of that name. And it usually begins with a lowercase. And generally when we do write a genus and a species, or the binomial scientific name, then we write it in italics if we can. Sometimes a subspecies is included and it's an additional name applied to more specific groups. So for a human, a modern human, our scientific name, or our binomial name, is Homo sapiens. And if we add the subspecies in there, it's Homo sapiens sapiens. It basically means wise human. Here's some more examples of some scientific names for some organisms, such as the lion, which is Panthera leo, and the leopard, Panthera pardus. Notice they both belong to the same genus, but because of enough genetic differences between them, they have different species names. As I go back to this slide, the scientific name Homo sapiens sapiens distinguishes the modern humans from those that appeared approximately 250,000 years ago. Here's another subspecies. It includes the Homo sapiens neanderthalensis, which is the Neanderthals or cavemen that you may have heard of. Although it's pretty easy for most people to grasp the differences between most species, like lions and leopards, being both definitely cats but not the same species, biologists sometimes have difficulty finding a single definition for a species that can consistently be applied across all organisms. So, the most commonly used concept is called the biological species concept. This is a group of individuals that can interbreed and produce viable fertile offspring with one another. It means that their offspring are healthy enough to survive and they're able to have babies of their own. Obviously, the biological species concept is difficult to apply to fossils that we find or organisms with asexual reproduction, such as in bacteria. All in all, though, the biological species concept helps us understand why species are distinct from one another. What actually is it that makes us distinct? Well, one of the major things is our genes and the alleles that a species possesses. The gene pool is the total of the alleles found in all individuals of a species. Changes in the frequency of an allele in a gene pool can only occur within a biological species. So some examples of that are lions and leopards are separate species and cannot interbreed. And the allele for the lion's spotless coats arose and spread within lions. And the spotless allele cannot be transferred to leopards. What does this all mean? Well, various lines of evidence suggest that the common ancestor of lions and leopards actually had a spotted coat, but the allele that eliminated the spots from lions arose and spread only within the lions, but this allele is not found in leopards. The spotless allele has not been transferred to the leopard population because lions and leopards cannot interbreed. The spread of an allele throughout a species gene pool is called gene flow. Gene flow cannot occur between different biological species because pairing between them fails to produce viable fertile offspring. This reproductive isolation can take two general forms. They're referred to as pre-fertilization, which prevents fertilization from occurring, and post-fertilization, which fertilization does occur, but what's produced is called a hybrid, and that hybrid cannot reproduce. It's usually sterile. Our next slides will take a look at the specific mechanisms of what's called reproductive isolation, which isolates species from one another using the biological species concept. The pre-fertilization barriers include spatial or habitat reproductive isolation, behavioral reproductive isolation, mechanical reproductive isolation, temporal reproductive isolation, and gamete incompatibility. Let's take a further look. In habitat or spatial isolation, closely related individuals from different species do not come into contact with one another, such as in the picture here. These are some garter snakes, and one of the garter snakes lives in a watery environment, the other one lives in a dry environment, and these two would never get together because their habitats do not allow them to do so. So they would never get together in real life and mate with one another. In other words, they would never leave their own habitat, which has specific conditions, to go find another one from the other habitat to mate with. Another example is a polar bear from the Arctic and a spectacled bear from South America. They would never encounter each other in natural settings, as one is from the Arctic and the other one's from South America. In temporal isolation, it's all about timing or the readiness of when the individuals want to produce. It's usually different in different species. For instance, in the spotted skunks, when they're ready to mate, it's at different times of the year. Or plants with different flowering periods, they can't fertilize each other if they're not sending their pollen out during the exact same time of the year. So temporal, both of them start with a T. Temporal and timing. Temporal is all about timing. In behavioral isolation, there's ritual behaviors that prepare partners for mating. And if we have different species, generally those mating behaviors are different so that the organisms don't even recognize one another if there's a difference, or they won't accept one another if the mating behavior is not just right. Such as these blue-footed boobies from the Galapagos Islands. They have a certain dance and some other behaviors that they do, dancing, showing the blue feet, putting their wings in the air, and giving each other twigs. If this isn't done properly, they wouldn't accept the other individual as a viable mate. So any little change in genetics that makes a mating behavior different can isolate species from one another. In mechanical isolation, sometimes the sex organs are incompatible between the different species, so the sperm cannot reach the egg, or they can't physically do the act of sexual reproduction. Many insects have what's called the lock and key type of genitalia, which is physically going to prevent sperm from contacting eggs if it's a different species. Or in the snails that we see here, the way the shells actually open is different in certain individuals or species, and they can't physically perform the act of sexual reproduction because of those physical differences. In gametic isolation, there might actually be an attempt to have sexual reproduction, or in the case of these sea urchins, release their eggs and sperm into the water where they could possibly meet up. But the proteins on the surface of the cell membranes of the egg and the sperm are different enough so that they can't bind together, and therefore fertilization cannot happen. It doesn't, as we know, it doesn't take much for a mutation in a protein to occur in DNA to change those proteins. So a little change in gametes can separate species from one another that way. Next, let's look at the post-fertilization barriers. We have hybrid inviability, or also known as reduced hybrid viability, hybrid sterility, or known as reduced hybrid fertility, and hybrid breakdown. Taking a look at those, we already covered the gametic in this picture, but moving on, that maybe the species are very close. They haven't diverged from one another that long ago, and so they're still able to have the act of such as sexual intercourse or sexual reproduction, and fertilization does occur. Well, what happens next? In some cases, in reduced hybrid viability, offspring is created. But the zygote might be so weak that it cannot complete development because of genetic instructions that are incomplete or in conflict. A sheep crossed with a goat can produce an embryo, but the embryo dies in the early developmental stages. Or in this salamander, this particular species of salamander, when they are hatched from the egg, they're so weak that they die soon after. They never reach reproductive age. In some cases, it's called reduced hybrid fertility, offspring that's viable are made. They are strong and able to survive, such as in the case of a horse and a donkey getting together to make a mule. But generally the mule is sterile or infertile. A mule is a hybrid that often we humans take advantage and actually make happen by mating horses and donkeys together, knowing that the result will be infertile. The hybrid created is infertile, but we do it because mules are very strong animals and very useful in farming and other industries, agriculture. They're very sure-footed on mountain trails. They're very disease resistant. And so we actually do that on purpose as humans, but the outcome is the infertile mule. This hybrid sterility often occurs because the hybrid individuals cannot produce viable sperm or egg cells themselves. Remember that during production of egg and sperm, homologous chromosomes must pair up and separate during the first cell division of meiosis. Because a hybrid forms from the chromosome sets of two different species, in this case the horse and the donkey, who are different species, its chromosomes are not homologous and thus cannot pair up correctly during the process. In the case of mules, the horse parent has 64 chromosomes and then therefore produces eggs or sperm with 32 chromosomes. The donkey parent has 62 chromosomes, so it produces eggs or sperm with 31. The mule will therefore have an odd number of chromosomes, 63, and no way to sort these into pairs during the first division of meiosis. Although a tiny number of female mules surprisingly have produced offspring, this event is so rare that the gene pools of donkeys and horses have remained separate. In the last one, in hybrid breakdown, viable fertile offspring are created, but future generations fail to produce offspring. So it all breaks down in the grandchildren, and the line then would end. This is the case in this type of plant pictured here, type of wheat. This slide illustrates the horse and donkey problem with their gametes. So a mule is a hybrid of a horse and a donkey. A hybrid is the live offspring produced by the mating of two different species and often sterile. Why can they still mate? Well, they share a common ancestor, and the common ancestor that they diverged from was quite recent. So they separated into their two different species quite recently in Earth's history. Given the definition of a biological species, it's clear that all humans belong to the same biological species. To understand the concept of races within a species, however, we need to first examine how species form. According to the theory of common descent, all modern organisms descended from a common ancestral species. The evolution of one or more species from an ancestral form is called speciation. For one species to give rise to new species, 3 steps are necessary. First, there must be isolation of the gene pools of subgroups or populations of the species. So somehow species must be isolated from one another into 2 separate groups. Then evolutionary changes in the gene pools of one or both of the isolated populations must happen, such as by mutation or by natural selection or other events that occur in one group but not in the other. Then the evolution of reproductive isolation between the populations. So we can't have gene flow to be able to share alleles back and forth with the 2 populations. So if there's isolation between them, enough differences build up over time. If we were to bring them back together again, they would no longer recognize each other, and we would have some of these reproductive barriers such as the habitat, temporal, behavioral, mechanical, or gametic isolation occur, that they wouldn't be able to mate with each other anymore and produce viable fertile offspring. They may want to, but in the case of maybe gametic or mechanical isolation, they wouldn't be able to. Isolation and divergence of gene pools may occur due to, well, migration. A small population might become isolated from the main population in a location far from them. Or there may be a geological barrier that separates them, such as a sudden change in a river's course separating 2 populations from one another, such as maybe some populations of squirrels get separated by a river. They don't swim and they don't ever go back and share alleles with one another. We would say there's no more gene flow now between the 2 populations because of the river between them, and now changes in alleles occur between the 2 different populations, and they're not able to share those alleles anymore. Enough differences build up over time, and they become different species. So migration, again, is the movement of a new population to a new location. They leave their main population and go somewhere else. An island bird, a reptile, a plant, an insect species seem to descend from a mainland species such as the Hawaiian silversword and Hawaiian honeycreepers and marine iguanas. They end up from the mainland, and they end up isolated from that group, and they're on their own island. Adaptive radiation may occur. Adaptive radiation is the establishment of a new population far from an original population, and individuals from a species move into new niches and start using those new niches. Natural selection occurs, and differences can build up, changing the species from what the mainland species looks like. Obviously, natural selection on the island, incidences there are going to be different usually than on the mainland. Let's look at some other isolation and divergence of gene pools. In allopatric populations, they're isolated from each other by distance or a geographic barrier, like this squirrel species being isolated by the Grand Canyon. The Isthmus of Panama separated members of aquatic species there, and over time the separated members became different species. So genetic evidence, if we look at that today, shows divergence from an ancestral species. In sympatric populations, the gene pools are separated despite the fact that they actually live together or near each other. They're reproductively isolated for other reasons, such as timing of reproduction or maybe a chromosomal change, which often happens in plants. And then plants, can actually reproduce asexually, so they can make more of themselves with their different number of chromosomes, but they can't breed back to the original population of plants that's around them. Here's another example of sympatric speciation in North America. This appears to be the case in populations of the apple maggot fly. They're notorious pests of apples grown in the northeastern North America area. However, the apple trees are not native. They were first introduced to the continent about 300 years ago or so. Apple maggot flies also infest the fruit of hawthorn shrubs, a group of species that are native to North America as well. Even though apples and hawthorns live in close proximity, flies from these two plants actually have little opportunity to mate because the fruits of these plants mature at different times of the year. As a result, there appears to be little gene flow between the apple-preferring and the hawthorn-preferring populations of maggot flies. Because there's no gene flow or little gene flow, the two groups of apple maggot flies have begun to diverge from one another. The gene pools of the two groups now differ strongly in the frequency of some alleles. Although not yet considered separate biological species, these two populations are on their way. They may continue to diverge and eventually become reproductively incompatible. Then, according to the biological species concept, they would be considered different species. In this chart, we see the isolation and divergence of the gene pools of these two different, or these apple maggot flies that have different preferences. The ones that prefer the apples and the ones that prefer the hawthorns. In plants, isolation of gene pools can occur instantaneously without any barriers between the populations, as I just recently said. A simple hybrid between two plant species is typically infertile because it can't make gametes. Same problem that occurs in mules, right? But some hybrid plants can become fertile if a mistake during mitosis produces a cell containing duplicated chromosomes. The process of chromosome duplication is called polyploidy, and it results in a cell that contains two copies of each chromosome from each parent species. If polyploidy occurs inside a plant bud, all the cells of the branch that arises from that bud will be polyploid, containing two identical copies of every chromosome. Because polyploid cells now contain pairs of identical chromosomes, meiosis can now proceed, and thus flowers produced on the branch can produce eggs and sperm. A polyploid flower can then self-fertilize and then give rise to hundreds of offspring, representing a brand new species that's isolated from the parent plants around it. Research also suggests that this process of instantaneous speciation may have been a key factor in the evolution of as many as 50% of flowering plant species. Polyploidy occurs in some animal groups as well, such as insects and frogs. In the picture, we see the plants kale with 18 chromosomes and turnips with 20 chromosomes. A kale egg would have 9 chromosomes, and a turnip egg, or turnip pollen, I should say, which is like sperm, would have 10 chromosomes. If they were to get together and make a hybrid offspring, then there would be an uneven amount with 19 chromosomes. But this is how we get canola. From polyploidy, the mistake during mitosis happens and leads to chromosomes duplicating twice. When the cell divides, its offspring can produce a branch with twice as many chromosomes as the rest of the plant. So now the branch has 19 pairs of chromosomes. Meiosis and self-fertilization proceeds, and our new polyploid species, or canola, is formed. To become truly distinct biological species, diverging populations must become reproductively isolated, either by their behavior or by their genetic incompatibility. In the case of canola, genetic incompatibility occurs immediately. A cross between canola and kale does not result in offspring. In most animals, the process of divergence may be more gradual, occurring when the amount of divergence has caused numerous genetic differences between the two populations. There's no clear rule about how much divergence is required. Sometimes a difference in a single gene can lead to incompatibility, whereas at other times populations demonstrating great physical differences can produce healthy and fertile or viable offspring. Exactly how reproductive isolation evolves on a genetic level is still unknown, and it's an actively researched question in biology. What we do know, however, is that once reproductively isolated, species that derived from a common ancestor can accumulate many differences, even completely new genes. When Darwin was around, he assumed or hypothesized that speciation occurred over millions of years as tiny changes that gradually accumulated, something that a human would never see in their lifetime. That hypothesis is known as gradualism. Other biologists have argued that most speciation events are sudden. They result in dramatic changes in form within the course of just a few thousand years, and are followed by many thousands or millions of years of little change. This hypothesis is known as punctuated equilibrium. The hypothesis of punctuated equilibrium is supported by observations of the fossil record, which seems to reflect just this pattern. Although the tempo of evolutionary change may not match Darwin's predictions, the processes of natural selection he described can still explain many instances of divergence. Regardless of the tempo of speciation, the period after the separation of the gene pools of the two populations, but before the evolution of reproductive isolation, can be thought of as a period during which races of species may possibly form, as described in our next section. So let's take a look at biological race. Biologists do not agree on a standard definition of biological race. In fact, not all biologists feel that race is even a useful term at all. Many prefer to use the term subspecies to describe subgroups within a species. However, the story that opened the chapter leads to a specific definition. That is to say that Simone Manuel's race influenced her likelihood of being a competitive swimmer presumes that when an individual is identified as a member of a particular race, that means she is more closely related and thus biologically more similar to individuals of the same race than she is to individuals of other races. This definition of biological race describes races as populations of a single species that have diverged from each other as a result of isolation of their gene pools. Some biologists may refer to this definition as the genealogical species concept because it reflects closer shared ancestry or a genealogy among certain individuals within a biological species. With little gene flow among these isolated groups, evolutionary changes that occur in one may not occur in another. Biologists use the genealogical species concept when arguing that isolated populations of an endangered species should be preserved as a way to preserve the unique genetic characteristics found within that species. To understand if the racial categories people in the United States identify with have a biological basis, that is, that they encompass gene pools that have long been isolated from each other, we must first understand the origin of our racial classifications. So here's a question: which species concept may be defined as biological race? That would be a genealogical species. Until the height of the European colonial period in the 17th and 18th centuries, few cultures distinguished between groups of humans according to shared physical characteristics. Historians of ancient Greece and Rome tell us that people of those eras primarily identified themselves and others as belonging to particular cultural or social groups with different customs, diets, and languages, regardless of their physical appearance. As Northern Europeans began to contact people from other parts of the world, setting groups of people apart made colonization and slavery less morally troublesome for the oppressors. This belief that human races were in fact biologically different from each other affected even scientists. Using the biological species concept, Linnaeus correctly classified all humans as one species. However, he also distinguished what he termed the varieties, what we'll now call races. The varieties of humans describing both physical characteristics and particular behaviors and aptitudes for each. Linnaeus did not have any biological evidence to support this notion of distinguishing human types, but the prevailing European attitudes about the natural superiority of their group over others influenced him, and even other scientists of the day, to describe the European variety as a naturally superior form. Linnaeus's classification of human races is an example of how scientists' social context has influenced their hypotheses. In this case, as in many others, this influence feeds back to society by seeming to justify and excuse injustice and brutality. Scientists since Linnaeus continued to propose hypotheses about the number and characteristics of races of the human species. Some species have described as many as 26 different races of human species, and some personal genotyping services, companies which analyze the genes of individuals to determine their ancestry, claim to be able to distinguish more than 50. But the most common number of hypothesized races is 5, as is still reflected in the race categories listed in the 2010 U.S. Census form. They are white, black, Pacific Islander, Asian, and Native American. However, do these groups truly represent biological races? To answer that question, we can try to determine if the physical characteristics used to delineate the 5 supposed human races, skin color, eye shape, and hair texture, for instance, developed because these groups evolved in isolation from one another? The data needed to answer that question comes from the fossil record and from the gene pools of modern populations. The ancestors of humans are known only through the fossil record. We cannot delineate fossil species using either the biological or genealogical species concepts. Instead, paleontologists, those are scientists who study fossils, they use a more practical definition. They use the morphological species concept. This is a group of individuals with some reliable physical characteristics, distinguishing them from all other species. It's more practical for paleontologists studying fossils to use this, as opposed to the biological species concept. The morphological differences among species are assumed to correlate with isolation of gene pools. Using the morphological species concept, that is, using reliable physical characteristics, scientists have identified the fossils of our direct human ancestors. This has allowed them to reconstruct the movement of humans since our species' first appearance on Earth. This table compares the three species concepts that we've looked at so far: the biological species concept, the genealogical species concept, and the morphological species concept. Again, in biological species concept, a species consists of organisms that can interbreed and produce fertile offspring that are reproductively isolated from other species. It's useful in identifying boundaries between populations of similar organisms and is a relatively easy in evaluating for sexually reproducing species. However, in disadvantages, it cannot be applied to organisms that reproduce asexually or to the fossil organisms that we've found. It may not be meaningful when two populations of the same species are separated by large geographical distances. In the genealogical species concept, a species would consist of organisms that can interbreed, are all descendants of a common ancestor, and represent independent evolutionary lineages. It's most evolutionarily meaningful because each species has its own unique evolutionary history and can be used with asexually reproducing species. But it's difficult to apply in practice. It requires detailed knowledge of gene pools of populations within a biological species and cannot be applied to fossil organisms. In the morphological species concept, a species consists of organisms that share a unique set of physical characteristics that's not found in other groups of organisms. It's easy to use in practice on both living and fossil organisms. You only need a few key features for identification, but it does not necessarily reflect evolutionary independence from other groups. The immediate predecessor of Homo sapiens was Homo erectus. That species first appeared in East Africa about 1.8 million years ago, and then spread to Asia and Europe over the next 1.65 million years. Fossils identified as early Homo sapiens appear in Africa in rocks that are approximately 250,000 years old. The fossil record shows that these early humans rapidly replaced Homo erectus in Africa, Europe, and Asia. Most data and evidence supports the hypothesis that all modern human populations descended from these African Homo sapiens ancestors within the last few 100,000 years. One line of evidence supporting this is that humans have much less genetic diversity, measured by the number of different alleles that have been identified for any one gene, than any other great ape, indicating that there's been little time to accumulate many different gene variants. Using the same reasoning, we know that African populations must be the oldest human populations because they are more genetically diverse than others around the world. Thus, African populations are likely the source of all other human populations. Given the evidence of recent African ancestry, the physical differences we see among human populations must have arisen in the last 150,000 to 200,000 years, or in about 10,000 human generations. In evolutionary terms, this is not much time, actually. All humans shared a common ancestor very recently. Thus, the defined human races cannot be very different from one another. Even with little genetic difference among populations, our question about the meaning of race is still relevant. After all, even if two races differ from each other only slightly, if the difference is consistent, then it would hold true that people are biologically more similar to members of their own race than to people of a different race. We'll explore the evidence that refutes this notion in our discussions that follow. To determine if one race was truly isolated from other races, researchers can examine the gene pool of populations described as a single race. Remember that when populations are isolated from each other, little gene flow occurs between them. If an allele appears in one population, it cannot spread to the other. As a result, isolated populations should contain some unique alleles. In addition to finding unique alleles, researchers should be able to observe differences among isolated populations in the percentage of the population that carries the particular alleles. When a trait becomes more common in a population due to evolution, it's because the allele for that trait has become more common. In other words, evolution results in a change in allele frequency in a population. The study of the effect of evolutionary change on allele frequencies is known as population genetics. This figure illustrates the relationship between individual genotypes and a population's gene frequency in a simple case, a single gene with just two alleles. Here, 70% of the alleles in the population are dominant, or big A, and 30% are recessive, or little a. If populations are isolated from each other, evolutionary changes that occur in one population will not necessarily occur in another. These changes would show up as differences in allele frequency between the populations. So, a population isolated from the other, shown in our figure, might have the same two alleles, big A and little a, but a different ratio. Say 50% are big A and 50% are little a. With this understanding, we can now make two predictions to test a hypothesis of whether biological races exist within a species. If a race has been isolated from other populations of the species for many generations, it should have these two traits. There should be some unique alleles, and there should be differences in allele frequencies relative to the other races for some of the genes. We'll now turn to the evidence and discover that the hypothesis that human races represent independent evolutionary groups is not supported. Recall the 5 major human races described in the 2010 census: white, black, Pacific Islander, Asian, and Native American. Do these groups show the predicted pattern of race-specific alleles and unique patterns of allele frequency? Well, the convincing evidence is absent. There's a lack of consistent differences among groups, and most evidence indicates that the genetic differences between unrelated individuals within a race are much greater than the average genetic differences among races. The Human Genome Project has allowed researchers to scan the genome of thousands of individuals looking for evidence of alleles that are found in a single human group and not in other groups. The types of alleles that are most commonly used in the analysis are called single nucleotide polymorphisms, abbreviated SNPs or SNPs for short. A SNP is a single base pair in the DNA sequence of humans that can differ from one individual to another. Humans are remarkably similar, having identical gene sequences for 99% of our genome. The 1% of the genome where there's variability is primarily made up of SNPs. We can think of the different DNA bases found at a particular SNP site as different alleles. They're actually also used to understand disease susceptibility and other health traits as well that affect human health and well-being. However, because 99% of SNPs appear to be in parts of the genome that do not produce proteins, they may have little or no effect on evolutionary fitness and thus can be readily passed on to future generations. So, SNPs are useful for understanding ancestry, but they're neutral. They have no effect on the physical characteristics of individuals. Researchers have identified a number of SNP alleles that are unique to human populations. Most genetic ancestry testing companies identify 3 major groups: African, European, and Asian, which includes Native American. Within those larger groups, certain populations display unique SNP alleles that may help identify an individual's ancestry more specifically. However, it's important to remember that the overwhelming majority of SNPs have no effect on phenotype, and that no SNP allele is found in every individual in any population. In fact, among groups classified in the same major race, some populations may have no individuals who have a particular SNP allele that's considered just unique to that race. Here's a question, true or false? No alleles are found in all members of a race. That's true. What is true of SNP alleles may be more clearly illustrated with alleles associated with readily apparent phenotypes. For example, sickle cell anemia has long been thought of as a disease that mostly affects black individuals. This illness occurs in people who carry 2 copies of the sickle cell allele, resulting in red blood cells that deform into a sickle shape under certain conditions. The consequences of these sickling attacks include severe pain and heart, kidney, lung, and brain damage. Many individuals with sickle cell anemia do not live past childhood. Nearly 10% of African Americans and 20% of Africans carry 1 copy of that sickle cell allele, whereas the allele is almost completely absent in Europe and in Americans. However, if we examine the distribution of the sickle cell allele more closely, we see that the seemingly race-specific pattern is not so straightforward. Not all human populations classified as black have a high frequency of the sickle cell allele. In fact, the populations from southern and north central Africa, which are traditionally classified as black, this allele is rare or even absent. Among populations who are classified as white or Asian, there are some in which the sickle cell allele is relatively common, such as white populations in the Middle East and Asian populations in Northeast India. Thus, the sickle cell allele is not a characteristic of all black populations or unique to a supposed black race. In fact, scientists have not identified a single allele that's found in all or even most populations of a commonly described race, but not found in other races. Only a tiny number of SNPs have been identified as unique to a particular human racial group. And these are never found in all populations of the race or within every individual of a population. The hypothesis that human races represent independent evolutionary groups is not supported by these observations. Certain SNP alleles are useful in that they link individuals to particular population groups. This is due to geography and similar selective pressures acting on those individuals in an area, gene flow within the population, and being able to share alleles in that area. But if race is to be biologically meaningful, then the allele frequency of many SNPs and genes should be more similar among populations within a race than among populations of different races. But populations within a race are often as different as populations across races, and allele frequencies are not more similar within racial groups than between them. More variability exists within a race than average differences among races, and both fossil and genetic evidence show that human racial groups do not represent biological races. The figures shown here, they're listed by increasing frequency of a particular allele in the population. The color coding of each population group in each of the graphs corresponds to the racial category in which the population is typically placed. For example, at the top of figure 1311, or picture A, the Taiwanese population is categorized as Asian, and the Cree Indian population of eastern Canada is categorized as Native American. If the hypothesis that human racial groups have a biological basis was correct, then populations from the same racial group would be clustered together on each bar graph. But picture A shows the frequency of the allele that interferes with an individual's ability to taste the chemical called phenylthiocarbamide, or PTC for short, in several populations. People who carry 2 copies of this recessive allele cannot detect PTC. It tastes bitter to people who carry 1 or no copies of the allele. Note that populations within a single race, like Asian, vary widely by the frequency of this allele. Graph B lists the frequency of one allele for the gene haptoglobin 1 in a number of different human populations. Haptoglobin 1 is a protein that helps scavenge the blood protein hemoglobin from old, dying red blood cells. Again, we see a wide distribution of allele frequency within the race categories. What we see in the graphs and the figure is that allele frequencies for these genes are not more similar within racial groups than among racial groups. In fact, in both cases, the populations with the highest and lowest allele frequencies belong to the same race for these genes. There is more variability within a race than there are average differences among races. Even though certain SNPs can help us track an individual to a particular ancestral population, the pattern of diversity in other genes tells us these ancestral patterns are not evidence of deep biological similarities among populations within a racial group. In fact, the categories for human race fail to meet the criteria for identifying populations as consistently isolated from each other. Both the fossil evidence and genetic evidence indicate that the 5 commonly listed human racial groups do not represent biological races. The results of genetic ancestry tests on individuals in the U.S. typically reflect the truth that populations have not been truly isolated for the past 500 years of European and Asian immigration and the history of African slave trade. Recent research indicates that most African Americans have at least 20% European SNPs, and that 30% of white U.S. college students have less than 90% European SNPs. But the melting pot of the United States is not unique in human history. In fact, the evidence that human populations have been mixing since modern humans first evolved is contained within the gene pool of human populations. For instance, the frequency of the B blood group decreases from east to west across Europe. The allele that codes for this blood type apparently evolved in Asia, and the pattern of blood group distribution seen in our figure here corresponds to the movement of Asians into Europe beginning about 2,000 years ago. As the Asian immigrants mixed with the European residents, our alleles became a part of the European gene pool. Populations closest to Asia experienced a large change in their gene pools, whereas populations who were more distant encountered a more varied immigrant gene pool made up of the offspring between the Asian immigrants and the European neighbors. Other genetic analyses have led to similar maps. For example, one indicates that populations from the Middle East migrated throughout Europe and Asia about 10,000 years ago. The data from these kinds of mapping projects indicate that there are no clear boundaries within the human gene pool. Reproduction among human populations over hundreds of generations explains why the isolation occurred, or why the isolation required for the formation of distinct biological races never occurred. How does the frequency of the B blood group change as humans migrate from Asia? The frequency of the B blood groups decrease. So, why do human groups differ? As we've learned, human races are not true biological races. However, human populations do differ from each other in many traits. In this section, we explore what is known and why populations share certain superficial traits and differ in others. Natural selection can explain a lot of the differences. For instance, with the sickle cell allele, it's been found that individuals that live in an environment that has a high incidence of malaria are more likely to have the sickle cell allele. It's considered an adaptation. It makes it harder for the malaria parasite to enter the red blood cells of the individual and affect them if they carry the one copy of the sickle cell allele. Basically, it increases their evolutionary fitness. Natural selection has driven an increase in the frequency of this allele in just the susceptible populations. The protection that the sickle cell allele provides to heterozygote carriers is demonstrated by the overlap between the distribution of malaria and the distribution of sickle cell anemia. Another physical trait that's been affected by natural selection is nose form. The pattern of the nose shape in populations generally correlates to climate factors. Populations in dry climates tend to have narrower noses than populations in moist climates. A long, narrow nose appears to expose inhaled air to more moisture, thereby reducing lung damage and increasing the fitness of the individuals to drier climates. Among equatorial Africans, people living at drier, high altitudes have much narrower noses than do those living in the humid rainforest areas. Interestingly, our preconception about the biological nature of human race puts two populations of Africans, represented by our figure here, in the same race and explains differences in their nose shape as a result of natural selection and the area that they come from. But places also white and black populations into different races and explains their skin color differences as evidence of long isolation from each other. However, like nose shape, skin color is a trait that's strongly influenced by natural selection. Traits that are shared by unrelated populations because they share similar environmental conditions are termed convergent. Convergent evolution occurs when natural selection for similar environmental factors causes unrelated organisms to resemble each other. For example, the similarity in the shape of the white-sided dolphins and reef sharks as a result of convergent evolution. We know by their anatomy and reproductive characteristics that sharks are most closely related to other fish and that dolphins are more closely related to mammals. The pattern of skin color in human populations around the globe also appears to be a result of convergent evolution as well, in which unrelated human populations appear similar as a result of evolution in similar environmental conditions. When scientists compare the average skin color in a native human population with the level of ultraviolet or UV light to which that population is exposed, they see a close correlation. The lower the UV light level, the lighter the skin, regardless of the race in which the population is classified. UV light is high energy radiation in a range that's not visible to your human eyes. Among its many effects, UV light interferes with the body's ability to store the vitamin folate. Folate is required for proper development in babies and for adequate sperm production in males. Men with low folate levels have low fertility, and women with low folate levels are more likely to have children with severe birth defects. Therefore, individuals with adequate folate have higher fitness than those without. Because darker-skinned individuals absorb less UV light, they have higher folate levels in high UV environments than light-skinned individuals do. In other words, in environments where UV light levels are high, dark skin is favored by natural selection. Human populations in low UV environments face a different challenge. Absorption of UV light is essential for the synthesis of vitamin D. Vitamin D is crucial for the proper development of bones. Women are especially harmed by low vitamin D levels. Inadequate development of pelvic bones can make giving birth deadly. There is no risk of not making enough vitamin D when UV light levels are high, regardless of your skin color. However, in areas where levels of UV light are low, individuals with lighter skin absorb a larger fraction of the UV light available and thus have higher levels of vitamin D than those with darker skin. So, in less sunny environments, light skin has been favored by natural selection. Because UV light had important effects on human physiology, it drove the evolution of skin color in human populations. Where UV light levels are high, dark skin is an adaptation, and populations became dark skin. You see more and more of those alleles in that population. Where UV light levels are low, light skin was an adaptation, and more people had that type of allele for lighter skin as the populations evolved. The exception to these patterns actually support the hypothesis. The relatively dark-skinned Inuits of the northern polar regions did not experience strong natural selection for lighter skin color thanks to vitamin D-rich fish diets. As you can observe in the pictures, the pattern of skin color in human populations is a result of convergent evolution of different populations in similar environments. Generally darker skin near the equator on each continent and lighter skin near the poles, not necessarily evidence of separate races of humans. Natural selection has caused differences among human populations, but it also has resulted in some populations superficially appearing more similar to some other human populations. In contrast, populations that appear on the surface to be similar may be quite actually different by chance. Genetic drift is a change in allele frequency that occurs due to chance. Human populations tend to travel and colonize new areas. So these small population groups are very prone to evolution by genetic drift or chance events that would affect their allele frequencies. There's two different situations at work here. One is called founder effect, or bottleneck effect, and then chance events in small populations. Genetic differences can occur when a small sample of a larger population establishes a new population. The gene pool of the immigrants is rarely an exact reflection of the gene pool of the main or source population. It depends who all became part of this new small population. That difference leads to what's called founder effect. Let's say in the case of the pictures here, we have a frequency of a red allele that's low in an original population. And then several travelers or survivors happen to carry the red allele, colonize a new area. So now the frequency of the red allele is much higher than it was in the old population. So as you can see, this skews the allele frequency. This is the case for the rare genetic disease Ellis-Van Creveld syndrome. The Amish of Pennsylvania are descended from a population of 200 German founders who immigrated to the U.S. more than 200 years ago. Ellis-Van Creveld syndrome, a recessive disease that causes dwarfism, among other effects, is 5,000 times more common in the Pennsylvania Amish population than in the other German American populations. This is because of the founder effect. Plants with animal dispersed seeds appear to be especially prone to founder effect. For example, cocklebur, a widespread weed that produces hitchhiker fruit designed to grab onto fur of passing mammals, consists of populations that are quite variable in size and shape. The variation among populations appears to have been caused by differences in the hitchhikers that happened to be carried to new locations, where they founded new populations. A variant of founder effect is called bottleneck effect, in which a disaster wipes out most of the population, leaving only a small subset of survivors. In this case, again, what if the original population had a low amount of the red allele, but then in the chance event where there was a disaster, it wiped out a lot of individuals but left a lot of the red ones, and now our new population after the bottleneck has more of the red allele and therefore a higher frequency of that red allele. So again, bottleneck also affects the allele frequency of the population when comparing the new population to the old population. So with chance events, small populations are especially prone to loss of alleles through chance. When an allele is in low frequency within a small population, only a few individuals carry a copy of it. If one of those individuals fails to reproduce, or if it passes on only the more common allele to the surviving offspring, the frequency of the rare allele may drop in the next generation. If the population is small enough, even relatively high frequency alleles can be lost after a few generations by genetic drift. A human population that illustrates that effect is a small population of the Hutterites, a religious sect with communities in South Dakota and Canada. Modern Hutterite populations trace their ancestry back to 442 people who migrated from Russia to North America between the years of 1874 to 1877. Hutterites tend to marry other members of their own sect, so the gene pool of this population is very small and isolated from other population. Genetic drift in this population over the past century has resulted in a near absence of type B blood among Hutterites, as compared to the frequency of 15 to 30% in other European immigrants of North America. Humans are a highly mobile species, and we've been founding new populations for millennia. Most early human populations were also probably quite small, and these factors make human populations especially susceptible to founder effect, bottleneck, and genetic drift, and have contributed to the differences among modern human groups that we see today. In addition to natural selection and random genetic change, humans' highly social nature may have enhanced certain superficial differences in appearance among different populations. Men and women within a population may have preferences for particular physical features in their mates. If this is expressed over generations, it can cause populations to differ in appearance. When a trait influences the likelihood of mating, that trait is under the influence of a form of natural selection called sexual selection, and can explain many male and female differences in animal species, such as the difference between the great plumage of a peacock versus the much lesser plumage of a peahen. And it can also account for differences in the human male and female traits of body size. You may ask, why are females choosing certain traits in the males that they mate with? Well, Darwin asked the same thing and explained it by, when a selection is made, it's an unconscious thought of choosing an individual to mate with who's very fit, such as a peacock who has a big, huge, colorful plumage would only be able to carry that and get away with it if they were a very fit male. So that would make them more likely to get more mates that way, and over time we would see more and more individuals in that population carrying the alleles for the greater plumage. In humans, there's some evidence that the difference in overall body size between men and women is a result of sexual selection, namely a widespread female preference for larger males, perhaps again because size may be an indication of overall fitness. However, some human traits may reflect simply a social preference. For example, some scientists have suggested that lack of thick facial and body hair in some human populations resulted from both men and women for less hairy mates. Although intriguing, there is as yet little definitive evidence to support that hypothesis, that sexual selection shaped any particular human trait, and no way to simply test it yet. Differences between populations may be reinforced by the ways in which individuals choose their mates. For instance, eastern bluebirds prefer to choose a mate who's just as colorful as themselves. This is called assortative mating, the preference to mate with someone like yourself. In humans, there's a tendency for people to mate assortatively by height, so tall women tend to marry tall men, and by social factors such as religious preference or education level. So this tends to exaggerate physical differences between groups. Although human populations may display differences due to natural selection, in certain environments, genetic drift, sexual selection, and assortative mating, the genetic evidence indicates that many of these differences are literally no more than skin deep. If human races are not biological real, is Simone Manuel wrong to describe herself as a black swimmer? No, because race is a social category, not a biological one. This distinction is illustrated by another athlete, the man who is often credited with being the first African-American gold medal swimmer of either sex, Anthony Irvin, who won his first Olympic medal in 2000. Despite his designation as first, the light-skinned, green-eyed Irvin, child of an African-American father and a European Jewish mother, does not identify himself as a black individual. He told Rolling Stone magazine, "I don't know a thing about what it's like to be part of the black experience, but now I do. It's like winning gold and having people ask you what it's like to be black. That is my black experience." Because Irvin did not appear to others as black, he did not experience the same concerns about his capabilities as a swimmer that Manuel faced. And he also did not come from a family where swimming was an unusual activity. In reality, black swimmers are not rare. Because African Americans are biologically less able to succeed at swimming, they are rare because pools and beaches in the United States were off limits to black swimmers for generations. Now that those social barriers have lifted, and more women, like Manuel, are demonstrating their excellence, it may not be long before the title black swimmer, just like black baseball player or black president, will be archaic, making a distinction that is not based in either biology or in social expectations. Here's some questions for us. A volcano eruption kills most of a squirrel population. The surviving squirrels have a different gene pool from the original population. What kind of genetic drift is this? That's bottleneck effect. Also shown on this slide, you'll see the front cover of a special issue of Time magazine issued quite a while ago, but showcasing America as quite the melting pot of a multicultural society of many immigrants. This computer image of a girl shows what maybe an average American may look like someday. As you finish our chapter, be sure to review the learning outcomes. Find them in your reading, review them in the lecture, and in your notes.